Hello and welcome to Science Research Weekly, Season 5, Episode 8. Cellular Senses and Psyche's Interior. Get ready for cellular mechanosensing, modeling the interior structure of Psyche, microbial succession in soil restoration, and Bayesian linear regression in R. This video covers material from March 13th to the 19th. Let's get started with some research articles. All right, first up is Emergent depth-mechanosensing of epithelial collectives regulates cell clustering and dispersal on layered matrices. So it turns out cells, even single cells, can sense the microenvironment, especially stiffness, looking through matrix layers. But in this current study, they actually found that collectives of cells, specifically epithelial cell collectives, can mechanosense matrices actually quite deep, up to 100 micrometers. And so some of this has relevance for things like cancer biology, because how tumors might invade might depend on how well the tumors sort of sense the environment and sort of, I guess, sense where they could invade, through what sort of matrix. All right, continuing the theme of matrices, we have this article exploring the interior structure of 16 Psyche through Basin-Scale collisions. So asteroid 16 Psyche is the largest member of what's called an MX-type asteroid, and actually might be a core of a planetesimal. And so in like 2029, 2030, NASA's going to fly by it to actually see if it's actually metal rich. But before then, through, I think, modeling, the researchers are trying to determine if they can figure out whether it has a metallic core, or it's a mix of iron and rock, or even just a primitive unmelted body, to figure out like what the interior matrix of materials is of Psyche. All right, and finally we have, for our normal articles, organic transparent and flexible films in exhibiting white light emission via Polymer-Network engineering and non-dye-centric strategy. So white light emitting materials are useful in engineering light applications, but they usually use fluorescent dyes to balance things. However, the matrix of whatever material isn't really used beyond hosting, but researchers are actually using the matrix to get sort of these better films that actually are really good at being transparent, flexible, and suitable for back elimination. So they're good candidates for next generation optoelectrical platforms. All right, we'll leave the standard papers and jump into the preprints. We have 3 from bioRxiv today. We have first, genome-wide discovery of cis-regulatory elements in a large genome. So cis-regulatory elements are often useful to explore, but it's hard to find them because these are non-coding regulatory elements, typically trial and error. However, researchers show kind of a couple ways to actually help sort of winnow down the locations to more easily find these. The first one is to doing bulk ATAC-seq to look at genome-wide patterns of chromosome accessibility in embryonic and adult tissues. And they're using the model system of, I think this is an amphipod. And then the second method they're doing is actually sequencing 3 very specific or closely related species of these amphipods to identify islands of sequence conservation across the genome. So there we go. Next up, we have taxonomic filtering accompanies functional expansion during long-term soil restoration. So researchers are looking at this grassland conservation project, looking at restored grassland. And while they found that sort of plant diversity recovered pretty quickly, the below-ground microbe community didn't, even after decades. It was sort of still not quite in equilibrium. That, passive regeneration isn't enough to have full soil functional recovery. You actually have to help targeted interventions to get the microbes sort of into equilibrium. All right, and final article here is Microscale spatial fragmentation promotes bacterial survival under antibiotics. So, unsurprisingly, at the small scale, microbes, where microbes are inhabit, there's fragmentation, and so if you're applying antibiotics, it's not going to be smoothly well mixed everywhere. And so actually the fact that the habits will fragment and there'll be different concentrations can actually predict how well bacteria will survive under antibiotic exposure. So that's an interesting sort of point of reality that you might not think about, oh yeah, small scale fragmentation actually will drive patterns of microbial persistence. And so that needs to be taken into account. All right, with that, we're done with the articles. We'll turn to some research tools. Just one today from our bloggers, from the R Programming Books blog. It's Bayesian Linear Regression R, a step-by-step tutorial. So if you're interested into Bayesian Linear Regression, this is a good way to step through and learn how to use this, including walking through different R packages you should know for Bayesian regression. And then finally, we have 3 new CRAN packages that might be of interest. We have SciProj, create a scientific project skeleton as an R package. There's causal plot, create publication-ready causal diagrams, and SciMeter, analysis of scientific publication data with R. With that, no new grants today, so we'll close here. Thanks for joining me for Science Research Weekly. Science on.